Today I'm going to talk about an unpublished side project that's been going on for a while, and that came out of our work on cellular stress in TDP and FUS inclusion formation. So, as you may know, a few years ago we have come up with this two-hidden model, which postulates that cellular stress plays an important role in TDP and FUS inclusion formation. So according to our model, which is summarized in this cartoon, the first hit or step in this pathological cascade is a nuclear import defect, which brings TDP or FUS into the cytosol. And then a second step is cellular stress, which leads to the formation of stress granules and the recruitment of TDP or FUS into these stress granules. And since we and others have found stress granule proteins in the mature inclusions of ALS and FTD patients, we hypothesize that these stress granules may be the origin of the mature pathological inclusions. Now, of course, this third step, so the conversion from reversible stress granules into mature inclusions, is still somewhat hypothetical, and we still don't understand the underlying mechanisms. So we got interested in studying this process in more detail, and to do so we used a very simple, so one idea that we had is that stress granule disassembly upon recovery from stress, so this reverse reaction here may be impaired, and we got interested in studying this in more detail. And a simple cell culture model that we used to study this process are HeLa cells that are stably expressing either FUS or TDP wild type, which are nuclear, or cytosolic mutants of the two proteins that have a defective nuclear localization signal, and therefore the proteins are partially cytoplasmic. So we subject these cells to a form of cellular stress, for example, heat shock for 1 hour, and then let the cells recover from stress. So in this case, we shift back the cells to 37 degrees, and then we can follow what happens during the stress recovery phase over time. Now, when we did these types of experiments, an interesting side observation came out, and that was actually something that we observed for the TDP wild-type protein. So this protein is, as you know, predominantly nuclear, so we don't really see it going into stress granules. But what we did see happening during this stress recovery phase is that nuclear TDP started to cluster in nuclear bodies or granules a few hours after heat stress. And this is shown in this time course here. So you see that, maximally 3 to 4 hours after heat shock, so after shifting back the cells to 37 degrees, TDP is really prominently localized in these nuclear bodies or granules. So we see the exact same thing for endogenous TDP-43. So if we use untransfected HeLa cells and stain them with a TDP antibody, you get exactly the same phenomenon, the same kinetics, and you see that it's a transient process, so 6 to 8 hours, and especially after overnight recovery, TDP is again redistributed homogeneously in the nuclear plasm. So we got interested in following up on this, not least because, this is the maximal time point. We got interested in this not least because it's been reported that occasionally there are also intranuclear aggregates of TDP-43 in some cases with TDP pathology, for example, cases with granulin or VCP mutations. So one of the first things that we checked was whether this is a TDP-43 specific phenomenon or whether it can be seen for other RNA-binding proteins. So we co-stained TDP-43 with other HNRMP proteins, but you see that none of the proteins that we tested, so FUS or HNRMP-A1, HNRMP-C, or HNRMP-M, shows the same behavior. So these proteins at the 4-hour recovery time point and also at other time points that we checked remain mostly homogeneously distributed in contrast to TDP-43. So it does seem to be somewhat specific for TDP-43. And so we're, of course, wondering what these nuclear bodies or granules are. So we looked up some reviews on nuclear bodies, I read about the different types of nuclear bodies that we could test. And we chose these 4 bodies as potential candidates. First of all, chiral bodies or nuclear gems, because it has been reported that TDP is partially localized with SMN in these nuclear gems. Then another potential candidate were, of course, nuclear speckles or splicing speckles, because they're storage sites for splicing factors. And a third obvious candidate were nuclear stress bodies, because they are described to form after cellular stress. And finally, from the number and size that our granules had, we also considered whether they could be maybe PML bodies, which are involved in, for example, in protein degradation. So we tested for localization to these different nuclear bodies by co-staining with specific markers of these nuclear bodies. And by that we could exclude that the bodies that we saw were chiral bodies or nuclear speckles or PML bodies. So I'm not going to show you this data. But what we could show is that we had very good colocalization with a marker of these nuclear stress bodies. It's called HNRP-associated, A1-associated protein or scaffold attachment factor B, so in short, SAF-B. So this RNA-binding protein, as you see here, shows pretty much the same behavior and kinetics as TDP-43 in the stress recovery model. And so since this is a good marker of these nuclear stress bodies, we think that this demonstrates that in the stress recovery phase, TDP is recruited into these nuclear stress bodies. And I should also mention that very similar findings were recently published by the group of Bob Balow. So if in case this data seems familiar to you, it may be from his published data in Human Molecular Genetics a few months ago, and our data are entirely consistent with their finding. So what are these nuclear stress bodies? So the function is not completely clarified, and the guy that has studied these bodies most intensely is Giuseppe Biamonti in Pavia, here in Italy. What his group has shown that these bodies are assembled on specific heterochromatic sites, namely arrays of satellite 3 sequences on certain chromosomes. So these are heterochromatic regions, but what happens after stress, for example heat shock, is that the heat shock factor 1 trimerizes and then becomes transcriptionally active and is recruited to these heterochromatic regions. And this in turn recruits a histone acetyltransferase, which decondenses these heterochromatic regions. And then Pol II can hop on and actually transcribe these satellite 3 repeat sequences into satellite 3 RNA. And these satellite 3 RNAs then recruit certain RNA-binding proteins such as SAF-B and TDP-43, as I've shown you on the previous slide. So these nuclear stress bodies are pretty much large ribonucleoprotein complexes that form then these very large clusters in the cell. And once the stress is released and sufficient levels of heat shock proteins have been synthesized, for example, HSP70, the HSF1 trimer is disassembled, and HSF1 and HAT fall off again, and also then these entire RNP complexes disassemble, and eventually the satellite 3 RNA is degraded, and the chromatin is again methylated, and heterochromatin is reformed, and the bodies are dissolved again. So, as I said, the function of these nuclear stress bodies is still somewhat hypothetical. So Giuseppe Biamonti has proposed that they change alternative splicing reactions during stress, because alternative splicing is, of course, regulated by the concentration of various splicing factors in the nucleoplasm. And so if certain splicing factors are recruited into these, transiently recruited into these nuclear stress bodies, their availability in the nucleoplasm is reduced, and then you get changes in alternative splicing. And our data and the group of the data from Bob Balow suggest that this may be the case for TDP-43 dependent splicing reactions as well. So since satellite 3 RNA has been shown to be important in the formation of these nuclear stress bodies, we tested if active transcription, so new RNA synthesis, is required for TDP recruitment into these nuclear stress bodies, and indeed it is. So if we add actinomycin D, so a transcriptional inhibitor, but not a translational inhibitor, to our cells to completely prevent new RNA synthesis, this will prevent localization of TDP-43 into these granules. So this is consistent with TDP-43 being recruited to these nuclear stress bodies. Now, to check how TDP-43 may be recruited into these nuclear stress bodies, whether this occurs via RNA binding or protein-protein interactions, we used a couple of TDP-43 mutants. First of all, the famous phenylalanine-interleucine mutation provided to us by Emanuele. So as you probably know, this mutant fails to bind to RNA, but what we could see is that at the 4-hour post-stress time point, this RNA binding deficient mutant still is perfectly normally recruited into nuclear stress bodies, as shown here. So the recruitment of TDP-43 into these nuclear stress bodies does not require RNA binding, so presumably it does not bind directly to these satellite 3 RNAs. And what we observed, and also what Bob Balow has already reported, is that the C-terminal region of TDP-43 seems to be essential, so this glycine-rich domain. So this is a construct that lacks the C-terminal region of TDP-43, and you see that in our cell recovery experiment, a stress recovery experiment, this mutant remains completely homogeneously distributed, so it fails to go into nuclear stress bodies. So recruitment of TDP into these nuclear stress bodies is clearly mediated by the C-terminal domain. And of course, one obvious question at this point was whether the ALS-associated mutations located in this region would somehow alter recruitment of TDP into nuclear stress bodies. But when we tested this hypothesis, to our disappointment, we found that none of the ALS-associated mutations that we studied showed any alterations in nuclear stress body localization. And this is also what Bob Balow has found, and what we could see for some artificial super mutants that we have created. Some postdoc in our lab has created. So he has combined a lot of different pathogenic mutations into one construct, and even these really severely altered super mutants show unaltered nuclear stress body recruitment. So it is still unknown how exactly TDP-43 via the C-terminal domain is recruited into these nuclear stress bodies, so which protein interactions are really responsible for this. And we did not so far go further to dissect this, because we are rather interested in the functional consequences of this. So does recruitment of TDP into these nuclear stress bodies impair the function of TDP-43? One of the most well-known functions of TDP-43 is, of course, the regulation of splicing, and it's relatively easy to test, so we decided to do this. and looked at several of the known published splice targets of TDP-43 and used their alternative splicing pattern as a readout for TDP-43 activity. So what we did is to isolate RNA at these different time points here. So the 4-hour recovery time point is the time point when TDP-43 is maximally localized in nuclear stress bodies, shown here in this red box. And you see that at this time point, we indeed do see an inhibition of, in this case, exon 3 inclusion, or here exon 2 exclusion. So we do see evidence of reduced TDP-43 splicing activity. As a control, we did the same experiment under conditions of TDP-43 knockdown, so with TDP-43 as siRNA. And from this, you can first of all see that these are clearly regulated by TDP-43, so they are true TDP-43 targets. And under these conditions, this shift that we observe here during this stress recovery time course is abrogated. And as a further control, we also looked at genes that are not regulated by TDP-43. So we chose a few genes reported by Clotilde Laguier-Torren and co-authors to be regulated by FUS, but not TDP-43. So these are 4 genes shown here. So in these control genes, we do not see a change in alternative splicing, in the alternative splicing pattern. So this does not seem to be a generic thing happening to all genes, basically, after stress. So with this, I want to end and to summarize that with this cartoon figure again. So we have hopefully shown that upon cellular stress, TDP is transiently recruited into nuclear stress bodies, and we think that this impairs its physiological function, for example, alternative splicing of TDP-43 target genes. So we think that cellular stress may not only be important for the formation of TDP-43 inclusions, but it may also functionally contribute to the TDP-43 dysfunction and changes in gene expression. So this work was still done in the lab of Christian Haas, and most of the experiments were done by a great technician, Claudia Abu Ayram, and these are other people in the lab of Christian who have also contributed to the project or are still working on this. I'd like to thank Emanuele for providing me with several constructs, and Manuela Neumann is a long-standing collaborator of ours on human pathology. And I'd in the end like to announce that I have in my new group, which I'll be starting next month, still 2 PhD positions available. So I'll be moving to the Institute for Anatomy Cell Biology of the LMU. And actually next spring we'll move to a brand new building, the new biomedical center of the LMU. And so if you have talented bachelor or master students that are interested in pursuing a PhD in cell biology of TDP-43 or FUS in Munich, please refer them to me. Thanks for your attention.